Hi, I'm Jonah Comstock, editor-in-chief of pharmaphorum. I'm here at ASCO 2026. It's the beginning of day one on Friday, and I am joined by David Spetzler, the president of Caris Oncology Solutions?
Caris Life Sciences.
Caris Life Sciences. And you are the president. You're also kind of the AI guy there, or one of the AI guys. And that's what I really want to focus on in this conversation, because I think that's going to be a big theme at the conference. You guys had your own big event last night. What was talked about there? What are you starting to hear now about the kind of some of the trends this year at ASCO, especially around the AI space?
Yeah, so I mean, this year is really when Agentic AI is coming into the scenes in terms of being able to access and understand genomic data. And so previously we were always reliant on machine learning type algorithms that were a little more prescriptive in nature. This year we now have agentic solutions where you don't have to be a mathematician, computer scientist, or bioinformatician to be able to start to query the data and gain insights very, very rapidly. And so we're able to find and produce signatures that are relevant to treatment decisions for very minute patient populations as well as large patient populations in a way that's just faster and easier than ever before. So it's a very exciting development in terms of our ability to extend life for patients. And at the end of the day, that's why we're all here, is how do we improve patient outcomes?
What's the role that Caris plays in the life sciences ecosystem? Are you working with pharma partners? Do you have your own assets?
Yeah, so we are the largest molecular profiling company on the planet. And so we're doing more sequencing of human genomic components than anybody else. And so our flagship product is an FDA-approved whole exome and whole transcriptome assay with companion diagnostic claims. And that's for solid cancer patients on the tissue. We have an LDT that is the same basic format, so whole exome, whole transcriptome out of the blood. And blood is very interesting because it allows us to start to track how the tumor is evolving over time, because it's much easier to get multiple samples of blood than it is tissue. One of the key components to blood testing, though, that really I think should be emphasized a lot is doing what's called chip subtraction. So in about 40% of late-stage cancer patients, there are mutations in their blood that look like oncogenes, that look like oncologically related mutations, but they're not coming from the tumor. They're coming from the immune system. And so the last thing you ever want to do is give a patient a cytotoxic agent for a mutation that's not coming from their tumor, because they still have all the side effects, but it's not going to cure their disease or impede their disease from further growth. And so chip subtraction is really, really important. And to my knowledge, we're the only ones that are doing that, certainly the only ones doing it at scale. And that becomes really, really important. And one of the great things about doing whole exome and whole transcriptome is the amount of data that we generate leads to a deeper understanding of the molecular nature of cancer. And so we've been able to leverage and utilize that data set to build a multi-cancer early detection test that has phenomenal early stage performance, because the way that we can truly bend the curve in terms of survival is really by finding cancer early, where we can surgically remove it and cure patients. And so identification of cancer at stage 0 or stage 1 is absolutely essential to really making a dent in terms of the mortality component to cancer.
Totally. I mean, I heard that ASCO has just changed the guidelines around colorectal cancer, where they're recommending a blood test to try to catch some of these earlier cases.
That's right.
So I expect we'll see more of that in the future as the blood tests get better.
A lot more. Yeah. And utilizing a technology that's appropriate to the biology really matters. So in colorectal cancer, for example, a lot of the cases are driven by changes in methylation. And so almost everybody is using a methylation-based approach to find these things in the blood. And it works reasonably well in colon cancer. Where it doesn't work well are in things like breast cancer and prostate cancer. Those are hormonally driven diseases. And so the approach that we've taken is actually ultra deep whole genome sequencing, because it spans the entire gambit of biological causes of cancer. And so we're in the early days of truly being able to use the technology that exists, because that technology is still limiting. So in a perfect world, we would have the sequencing capacity as a species to be able to do 10 to 20,000x whole genome sequencing. And it's just not there yet. We need a couple orders of magnitude improvement in the underlying machinery that we rely on for sequencing. But that's coming. The sequencing growth has been beating Moore's law. And so in 5, 6 years, we should actually be at that point in terms of having the technology to do what should be done and what could be done and what will be done in the future.
Is the long-term vision to have like a single blood test for cancer where it's just detecting all different kinds of cancer?
Absolutely. And it's not just cancer. So at the end of the day, when you think about the nature of disease are cells going wrong inside our body. And if the cell is going wrong, there's something at the molecular level that's gone wrong. And so by doing complete ultra deep whole genome sequencing, we're not just going to find the patterns that are indicative of cancer. We're going to find the patterns that are indicative of everything that is a chronic disease. So from Alzheimer's to rheumatoid arthritis to diabetes, at the end of the day, the molecular system is driving all of these things. We just need to find those signals. And those signals then are going to be super valuable, not just to diagnose the disease, but it tells us what type of drugs need to be built in order to inhibit them. And so this is the foundation of also incredible advances in therapeutic development as well.
When it comes to AI, have you found that sort of the rise of generative AI and agentic models and all this is commodifying the space, is giving you more competitors, or are there still kind of barriers to entry that let you kind of stand out because you've been doing this, you have the data, that sort of thing?
Yeah, so the agentic AI alone doesn't allow you to do much of anything. I mean, it's a convenient interface, so you can generate models without having to code, but it's the underlying data set that actually matters. And so we've been doing whole exome and whole transcriptome for years and years and years and have amassed this enormous data set to over 650,000 patients with whole exome and whole transcriptome and longitudinal outcome data. And so the agentic wrapper is just kind of the icing on the cake that sits on top of our enormous data set. But the true value comes from the data set itself. And so when you think about what is the journey in terms of how you go from data to actually something that matters, and there's a simple kind of paradigm. And so you can generate data. The key is to take that data and turn it into information, the information into knowledge, and then it's only when you act on the knowledge that it becomes wisdom. And so the goal is to go from data to wisdom as quickly as possible. And that's the excitement around agentic AI, is it speeds up that process. But you have to have that underlying data. And sequencing somebody today doesn't let you do anything because you have to wait and see, okay, what happened to them over the next couple of years. And so that clinical maturity is a really critical component in the development of these new AI-based signatures.
You talked about the promise of your technology in diagnostics at the point of care. What about on the research side? Are companies using your technology there?
Yeah, of course. So, one of the key attributes of, again, trying to go from that data to wisdom is being able to intervene in new ways. And so with such a complete data set, we're able to identify those patients that are resistant to existing therapies. That's the patient population where we need to develop new therapies. And so we can go in, identify, okay, this set of patients are resistant to existing therapies. What are the mutations that they're harboring that would enable us to develop new drugs to help that patient population? And so the beauty of our approach in being so comprehensive is that we're really able to service of the patient today and help them live longer, but at the same time generate the information and knowledge needed to help bring new drugs to market for the patient of tomorrow as well.
Well, thank you so much, David. This is really interesting stuff.
Thank you. Pleasure.